Hello, second year. Welcome to your Molecular Biology Period 1 post-lab discussion. So I opted for a recorded lecture video as I will be attending an important meeting later on. So pakiaral po muna tong lecture video na to and then we will proceed with our laboratory examination at around 2:30 or 3 p.m. I don't know which works best for you. So let us just start. So for our post-lab discussion, I was not able to discuss anything from period 1 na post-lab discussion. Hindi ako nakapag-discuss ng kahit ano. So let us just have a quick rundown of our laboratory experiments or laboratory simulations that we did on period 1. So dalawa lang naman yon. Plus, dinagdagan natin ng laboratory safety. And for that laboratory safety, we will be discussing the biosafety and biosecurity. So important si biosafety and biosecurity for molecular biology kasi isa siyang foundation or isa siya sa mga important trainings to be attended to before you will be able to work inside a molecular biology laboratory. Okay, so let us just discuss biosafety and biosecurity. So according to CDC, this is the sequence of how you will put on or what we call the donning of your personal protective equipment. So it may differ in different institutions, but according to CDC, ito yung basic. Number 1, you will wear your gown. Okay, the laboratory gown. Number 2, your mask or respirator na dapat snug fit. So hindi po mag-work ang isang mask or respirator kapag hindi snug fit sa ating mukha. Pagka medyo may nakalawit na ilong or nakalabas sa bibig or nakababa yung mask, it won't work the way it should work. Okay po, kasi nga ang goal is to protect our nose and our mouth from the droplets or aerosols. So dapat snug fit siya. Number 3, your goggles or your face shield, it must be placed over face and eyes to adjust to fit. And lastly, yung inyong mga gloves. But before anything else, dapat mag-perform muna tayo ng hand washing bago natin suot tong mga to kasi sterile po ito. So kung meron po tayong isusuot na bunny suit or meron tayong isusuot na shoe cover, so dapat parang ang suot muna is shoe cover muna and then paakyat. Tsaka may susuot si bunny suit or susuot si gown and then si inyong mask, yung goggles, di ba? From foot to head, parang ganun. And lastly, yung inyong gloves, outer gloves. May mga instances na magsusuot tayo ng double gloves. So, ang unang-unang mo natin susuotin is yung inyong shoe cover and then susuotin natin yung inyong first layer ng gloves nyo and then huling-huli yung outer gloves nyo. So, it will depend or it will vary depending on the institution. But as per CDC, itong apat na to yung dapat magkakasunod. Okay po? And as for the doffing or what we call how to safely remove your PPE, ito yung example. Siyempre, mag-uumpisa tayo sa pinakamadumi. According to CDC lang naman, si gown and then yung outer gloves. Pero kung tatanungin niyo po ako, and as per our training, ang pinakamadumi po talaga na inyong PPE is yung inyong shoe cover. Kasi yun yung pinanlalakad-lakad natin inside the laboratory. Next are your goggles or your face shield na dapat hindi matatouch ang ating skin. Dapat away from our body. Okay, kasi madumi na yan, contaminated na yan. Number 3, your mask or respirator. Meron pong proper way on how to remove the mask or the respirator. You should follow these following steps. And after that, mag-wash ka ng hands. Okay, or use even a sanitizer kapag hindi naman visibly soiled yung dumi sa inyong hands. Since naka-double gloves ka naman, na pwede naman na mag-work si hand sanitizer or alcohol. But pag medyo OC, pwede tayo mag-wash ng ating hands. Okay? Do not forget to perform the hand hygiene between the steps. If tatanggalin nyo itong gloves, okay, so mag-hand sanitizer. Tatanggalin si first layer of gloves, mag-hand sanitizer. Ganun po siya. Para hindi nako-contaminate. Contaminated na nga, mas mako-contaminate pa. Para ma-avoid yung cross-contamination. Okay, when working inside the laboratory, we have different types of waste categories. And each waste category should be thrown into proper location or places or proper color ng kanilang plastic bags. Hindi po sila pwedeng maghalo-halo. Pakitandaan po yan. So kung sa bahay natin, pwede natin gawin yun. Okay? Pwede natin paghaluin yung mga nabubulok sa hindi nabubulok, pero hindi naman dapat. When working inside the laboratory or when working inside the hospital, meron po tayong proper color coding scheme and waste categories. Again, for the black, sa ating black na plastic bag or anything, the waste category should be non-infectious dry waste. Yung mga pinagbalatan ng mga syringe, provided na walang blood doon, walang any touch ng blood, pwede po natin itapon doon sa waste categories, yung mga pinagkainan ng chippy, pinagkainan ng clover, basta walang nabubulok, pwede po natin ilagay sa ating black bag. Next is the green bag. It should be non-infectious wet waste, non-infectious wet waste, yung mga nabubulok na food or anything na wet, basta non-infectious, considered na non-infectious, pwede po sila sa green. Next, yung mga yellow bag. Tandang-tanda natin ng yellow bag, dapat yung mga infectious and pathological waste, dapat tinothrow sila sa ating yellow bag. Wag po kami makakakita pag nag-work na tayo sa laboratory natin sa face-to-face classes na ang mga barrel ng syringe ay itatapon sa black or sa green. Zero na po kayo sa activity. That particular activity, zero na po kayo. Okay po. And then the yellow with black band, it should be the chemical and heavy metal waste. Kapag naman nakakita kayo ng orange, usually hindi naman to sa laboratory, sa mga radiology department naman nakikita natin ang mga radioactive waste. And lastly, yung mga kulay red. Paminsan white yung kanilang katawan, yung parang pinaka-drum, plastic drum, ganun. Pero yung pinaka-ulo niya, yung pinaka-cover niya, red siya. Kasi it is under the waste category of sharps and pressured containers. Sharps meaning yung mga needle ng inyong mga syringe. Dapat doon po tinatapon. Hindi po sa yellow, hindi sa green, hindi sa black. Dapat sa red. Anything na sharp and has a pressured container, it should be thrown in the red bag. Okay? So next, this is the foundation of our molecular biology laboratory, the laboratory biosafety and biosecurity. This is one of the course now we've undergone or training we've undergone before I was able to work inside the molecular biology laboratory. So basic to, dapat pasado kami sa training ng biosafety and biosecurity, and after nun, nag-training na kami ng PCR hands-on. So after nung biosafety, biosecurity, ano yan? Parang nasa canvas kami. Parang may lecture part and then kailangan mapasa namin yung examination. Pag nakapasa kami doon, tsaka kami po proceed sa PCR training, hands-on, and meron ding lecture part. Okay? And then pag nakapasa na kami doon, nakapasa na tayo mga medtech doon, that will be the time na matatawag na tayong molecular biology analyst. So yung mga medical laboratory sciences, ang daming sections na pwede tayo kung saan mag-work. Isa na doon si molecular biology, and then before you became a molecular biology analyst, you should be able to pass these trainings first. So, hindi po basta-basta sinasalang lang tayo doon. Sobrang dami natin pagdadaanan. So, the laboratory safety, it is the containment principles, technologies, and practices implemented to prevent the unintentional or accidental exposure to pathogens and toxins or their unintentional or accidental release. So, ang daming principles underlying the biosafety para hindi kumalat or mag-spread yung mga diseases or yung mga contaminants na wino-work on natin inside the laboratory at hindi ma-acquire ng inyong general public. Ang dami, sobrang dami po. Hindi lang siya basta-basta na nagtrabaho tayo sa molecular biology laboratory. Sobrang dami. So, i-discuss na lang natin siya much further. Pagbabalik na tayo ng face-to-face classes, i-dissect natin yan isa-isa. Tuturuan ko po kayo. But for the laboratory biosafety, it is the containment principles or technologies or lahat ng mga practices na gagawin natin para ma-avoid natin ang unintentional or accidental release of these biohazards. Meanwhile, your biosecurity, it is the institutional and personal security measures designed to prevent the loss, theft, misuse, diversion, or intentional release of the pathogens and the toxins. So if you can notice, meron tayong main difference when it comes to differentiating your biosecurity and biosafety. Biosecurity, naka-highlight or naka-bold yung word na intentional. Gagawin natin ng lahat para ma-avoid natin yung intentional release ng mga pathogens and toxins or what we call yung mga bioterrorism. And dito naman sa biosafety, yung mga unintentional, ito yung mga good practices na ginagawa natin mga medtech. So yung mga pagsuot ng PPE, yung mga ganyan, under po yun dito. Pero pagka yung mga paglagay ng password sa pinto, yung pag-hire ng security, yung pag-isolate ng isang laboratory, ganyan. Under po sila ng biosecurity para hindi ma-release yung mga toxins and pathogens nung mga may intention na i-release. Okay, so kung may kagalit ka at gusto mong i-release yung pathogens para makaganti sa kanya, ayun po, it will fall under the laboratory biosecurity. Gagawin natin ng lahat para hindi mangyari yun. Okay? And then, again, the review of the biosafety and biosecurity. When we say biosafety, laboratory biosafety, it is keeping the people from the bad bugs. And meanwhile, the biosecurity is keeping the bad bugs from the bad people. And when we combine these two principles, biosafety plus biosecurity is equals to biorisk. Please do not forget. So again, ano ba yung mga examples na pwede nating ma-compare si biosafety and biosecurity? So meron tayong isang scenario dito where Mr. Blunder, I can't read his name, exposes the city to smallpox. Again, as we all know, eradicated na si smallpox. Meron man tayo niyan, hindi na siya cases. Nasa laboratory na lang siya. Ginagawa na lang, pinag-aaralan na lang yung smallpox virus. Hindi na po siya existing sa mga tao. Wala nang case ng smallpox sa mga tao ngayon. But Mr. Blunder was working in a laboratory, I think, and he accidentally exposed everyone in the community with the smallpox. It is unintentional because he did not know na meron na pala siya yung smallpox. He was able to acquire it in the laboratory and unintentionally he spread it in the community. Okay? And meanwhile, this person, si Diane Thompson, what she did was, parang ang nangyari is, yung E. coli, intentionally nilagay niya sa food ng kasama niya. Parang ganun, if I can remember correctly. Parang ganun. So, that was intentional. Meron siyang masamang intensyon na gumante dun sa ka-work niya sa laboratory. So, that is a big no-no. Kahit gaano kakagalit sa iyong kasama sa laboratory, wag na wag po natin gagawin yan. Okay? So, that is the main difference between biosafety and the biosecurity. Mr. Blunder did not know that he acquired the smallpox. And gagawin natin ng lahat. Okay, i-apply natin ang principles ng biosafety for here. But for the biosecurity, dapat pigilan natin si Miss Diane Thompson na gawin yung mga masasamang bagay na katulad nun. Kasi nga, intentional yung paglagay ng mga bacteria or specific type of pathogens cause harm to other people. So next, tapos na tayo sa basic concepts of the biosafety, biosecurity, and then alam niyo naman na ang laboratory safety basics, alam na yan. Let us just proceed with the DNA extraction. I know meron kayong activity for the cytogenetics na nag-discuss ng at gumawa ng experiment regarding the DNA extractions. Unahin natin si DNA extraction. So kung papanoorin niyo yung YouTube video na pinrovide ko ng laboratory tour, ito yung pinaka-ginagawa natin doon. So basically, magko-collect tayo ng specimen from our patients. As medical laboratory scientists, we are allowed to collect these samples from our patients. Parang authorized sample collectors tayo, parang ganun. So after natin makakollect ng, let's say, sputum, ng buccal samples, and nasopharyngeal samples, ang gagawin natin doon, i-extract natin yung DNA coming from that specific samples, and we would want to know kung anong type of virus, what type of pathogen, or what type of bacteria ang present dito. So that is why we are subjecting this into a DNA extraction on a molecular level to test what type of pathogens ang present doon. So kung manual extraction yung gagawin natin, let's say walang budget si laboratory or nag-start pa lang si laboratory, pwede po tayo mag-start sa basic, which is the manual extraction, at dadaan yan sa iba't ibang steps at medyo mahaba-habang steps. Siyempre, pag manual nga naman, compared to automated, mas mahaba yung steps niya. So for the manual extraction, parang ang mangyari dyan is, siyempre, cell harvest, cell lysis, protein removal, DNA binding, wash, hanggang sa makuha mo na yung elute mo. And then the usual principle behind the manual extraction is what we call the silica-based DNA extraction. So, ang ating manual, parang meron yan wash. Parang ano siya, wash, tapos in between may wash. So parang sobrang daming pagdadaan ng process. Although hindi naman tatagal ng tag-iisang oras dito, tag 15 minutes lang. But as medical laboratory scientists, we want na mas automated para hindi na tayo dadaan sa vigorous steps na to. Okay po? But for manual extraction, kung magsa-start pa lang, pwede po natin gamitin yan. Ano din naman to? Accurate din naman and very reliable. And goal is to get the DNA elute. So pag medyo may budget naman na yung ating laboratory, pwede na tayong mag-proceed with the semi-automated. Again, nasa DNA or RNA extraction pa rin tayo. If you will look closely, merong cartridge dito. So may mga butas yan dito lahat. May mga well, what we call the well. Meron yung mga reagent or may mga alcohol dyan in between. So, paano po ginagawa nitong machine na to para makakuha ka ng DNA from that sample? So, imagine mo yung nasopharyngeal sample mo nandito. Tapos, machi-check ko kung may makukuha bang certain type of virus. So, gagawin ito. Yan. Papanoorin po natin siya dito ha. I-click natin yung link later on. Pero kasi recorded video to. So, separate na lang natin. Gagawin niya parang bababa yung probe. And then magwa-wash. Parang ganun din. Parang dumaan din dito sa manual extraction. Basic principle. Ang difference lang, si machine na yung gumawa nun para sa'yo. Okay? Mas maganda ang ating mga semi-automated compared to manual kasi nga naman, it will take very much long time and mas mababa yung throughput mo. Ibig sabihin, mas konti yung samples na magagawa mo dito kasi nga isa lang naman kamay mo or mas mahaba yung time na iaalat mo para makakuha ng DNA samples. Okay? Mas okay pa rin naman si DNA semi-automated. Pero walang mas gaganda kay automated, fully automated. Ang gagawin na lang ni medical laboratory scientist dito is tatanggalin yung swab. Okay? For example, meron kang VTM, yung viral transport medium, yung ginagamit sa PCR, tatanggalin mo lang yung swab and then salang. And then the machine will do the work for you. Parang naka-chill na lang si medtech na nanonood doon. Okay? So next, yun lang naman ang basics ng ating DNA extraction. So ginawa nyo sa bahay nyo is parang manual DNA extraction using alcohol and soap ba? If I believe, ganun po siya. So next, let us just proceed with the polymerase chain reaction. So ang gagawin nyo, pag bumalik na tayo sa face-to-face classes, mag-uumpisa tayo sa DNA extraction. So pwede kayong kumuha ng mga samples dyan sa inyong classmates depending on our laboratory activity for that particular day. Uutusan ko kayong kumuha niyan, syempre. Tapos, sa DNA extraction, syempre, school lang tayo, manual. Mag-start tayo sa manual parati para alam niyo yung basics. And then, ang susunod na step nyo na doon is the polymerase chain reaction. Meron po tayong different types of polymerase chain reaction principles and machines as well. Pero ang available doon sa ating St. Jude College Manila machine is later on, discuss ko. So, polymerase chain reaction. What is polymerase chain reaction? As we all know, sometimes called the molecular photocopying. It is the polymerase chain reaction is a fast and inexpensive technique used to amplify. Pag sinabing amplify, meaning to copy a small segments of your DNA. It is a novel molecular training involving in vitro. In vitro, pag sinabing in vitro, it is the Latin word meaning outside of the human body. Pag in vivo, inside. Okay? Enzymatic replication of defined DNA sequences. And lastly, it is the often heralded as one of the most important scientific advances in the molecular. Today, PCR is used to diagnose diseases, identify bacteria and viruses, match criminals to crime scenes, and then the biotechnology. So, your polymerase chain reaction, meron siyang three basic processes na pinagdadaanan. Ano po ulit sila? Si denaturation, your annealing, and lastly, your extension or your elongation. Huwag pong kalilimutan. Yung polymerase chain reaction, again, is the in vitro enzymatic amplification of a specific segment of DNA. Theoretical basis was first described by Klepp in 1971, and then your double-stranded DNA denatures at high temperature. So kung mapapansin nyo, each step, meron silang different type of temperatures. Merong tataas, may bababa, and then mag-stabilize siya depending on the step. And then exponential increase of dsDNA produced with increasing cycles. Nabanggit na natin yung mga cycles na yan. So again, your equipment for PCR, usually, ito po. si conventional PCR, yung makikita natin sa St. Jude College, Manila na bagong bili, is what we call the conventional PCR machine. So again, start tayo sa DNA extraction. Kumuha kayo ng sample sa kaklase nyo. And then, sinubject natin siya into different washing hanggang sa nakakuha tayong DNA elute. Yung DNA elute na yun, lalagay natin sa parang well or PCR reaction tube. And then, babasahin siya dito sa ating thermal cycler. Okay, thermal cycler po yung kanyang principle. Ito po yung usual na meron tayo. Pagka medyo mura-mura pa ng konti or gusto nyo mag-start muna sa basic, pwede po natin gamitin si conventional PCR machine. Ito naman, ito yung maganda, si real-time PCR machine. Ang kinaganda nitong ni real-time PCR machine, hindi pa tapos i-read yung samples at around 4 hours, meron na siyang resulta. May kita mo dito sa graph. Pag may nag-amplify yung graph, ibig sabihin positive yung pasyente. Okay? Kung COVID yung or SARS-CoV-2 yung tinitingnan nating test. Pero kay conventional PCR machine, okay naman siya. Kaso ang difference lang is meron ka pang kasunod na step na gagawin kay conventional PCR machine. Dito kay real-time, after ng DNA extraction, template, ito na. Meron ka ng resulta, pwede na nating i-release sa pasyente. Kapag kay conventional PCR machine, ang kasunod mong step niyan is your gel electrophoresis. But your traditional or endpoint PCR use agarose gels. Okay, ito yung gel electrophoresis, kaya natin siya pinag-aaralan kasi sa St. Jude, i-apply natin siya sa face-to-face classes. Use agarose gels for detection of PCR amplification at the final phase or endpoint of the PCR reaction. Okay, so from the DNA extraction, sasubject nyo muna yung inyong kaklase or patient sa DNA collection. Okay, kakuha kayo ng sample, swab, or anything. Next, DNA extraction. Tapos, lalagay na natin siya sa ating PCR machine, conventional PCR machine. And then lastly, yung ating gel electrophoresis. Your gel electrophoresis is used to analyze, or it is solely mainly used for the analysis of PCR product. Your agarose gel electrophoresis, yun yung mas ginagamit natin. Your agarose gel, yung medium na mostly used for the molecular biology techniques. And then what does your agarose gel electrophoresis do? It separates DNA or RNA molecules by size. Again, it separates DNA or RNA molecules by size. It is achieved by moving negatively charged nucleic acid molecules through an agarose matrix with an electrophoretic field, or what we call the electrophoresis. Again, pag sinabing gel electrophoresis, the shorter the molecules, it will move faster towards the cathode. And then it will migrate farther. So kung naglagay ka dito sa sample well, mas mabilis makakapag-travel ang ating mga mas maliliit na DNA. Kasi po, merong mga filters yan, gel na yan, agarose gel na yan, meron siyang porous. It is porous, meaning madaming siyang filter. The DNA samples can move faster if they are shorter in nature. With the help of the buffer kasi naman din yan. And then yung ating saksakan, kailangan po yun. Okay? If the expected band is present, the PCR reaction is successful. Again pala, kapag ka magpupur ka ng iyong sample dito or yung inyong PCR product, mas maganda na samahan na natin siya nung mga intercalating dyes. Para nga naman, mas mas makita nyo siya, mas maganda yung visualization niya after on. Mas madali natin mababasa yung mga BP or yung mga base pairs natin afterwards. Okay? Maganda naman ang ating mga intercalating dyes to use. However, they are toxic and carcinogenic. So, you have to be very careful in working with your intercalating dyes. Okay? So again, your gel electrophoresis is the standard method to separate, identify, and purify nucleic acid fragments. And then the rate of movement of your DNA through gels is influenced by these several factors. So maraming mga interfering factors pa din, and then maraming factors that can affect the migration of the DNA. So we have to be very careful with that. Okay? So like what we've mentioned, dapat mag-work tayo accurately, and then we have to practice good laboratory practices para ma-avoid natin yung mga interfering factors, and then makakuha natin yung result na ini-aim natin. And then these are the properties of different types of your agarose. And then the strong sharp band indicates the uniform size and sufficient quantity of DNA, and then the smear indicates fragment of DNA sample. So, paano man po ito basahin? Tuturuan ko na lang po kayo later on. Okay? But basta ang importante, alam nyo na yung principle kung saan nag-uumpisa yung DNA extraction, DNA collection up until masubject na siya into gel electrophoresis. The interpretation will be taught later on na lang po. So that concludes our discussion for your post-lab discussion. I hope you learned a lot, and then you can apply this kapag nag face-to-face classes na tayo. So good luck on your examination. Thank you and good luck.